All right, so welcome to our first lesson of Unit 6. We're going to talk about DNA and RNA structure. Some of this is going to be review from when you were freshmen. Some of it is going to be a little more complicated. Let's dive in. So first, let's talk about chromosomes. Chromosomes are different in different organisms. Prokaryotes have a single circular chromosome. It tends to be much smaller. So on average, anywhere from zero point 0.6 to 10 million base pairs. Eukaryotes, on the other hand, have more DNA. They have multiple linear chromosomes, and it tends to be longer, anywhere from 10 to 100 million base pairs per chromosome. Because eukaryotic chromosomes are much longer, they have to be organized a lot more carefully to fit inside the nucleus and also to prevent tangling, DNA errors, etc. So eukaryotic chromosomes are highly organized. The chromosomes are condensed by wrapping around these proteins called histones. So here you see a group of histones. When those histones are then wrapped with DNA, that forms a unit called a nucleosome. These nucleosomes can then coil up, and then that ends up ultimately letting them form chromosomes that are visible during meiosis and mitosis. When chromosome is uncondensed, it's going to look a little different. And we're going to look at that on the next slide. You can actually use histones to regulate gene expression by modifying them, which makes them more or less unlikely to be unwound. So in some parts of the genome, you might have histones completely wound up around DNA much of the time, much of the lifespan of the cell, so that it's not able to be expressed, which then ultimately effectively turns off transcription of certain genes. So if we look at this, you have your histones can be either tightly wound around the DNA, which is this closed chromatin or heterochromatin. This type of chromatin you're not able to readily access and read. So that's going to be some DNA that's not going to be read easily. You're not going to have transcription occur there. On the other hand, this more loosely packed chromatin, this open chromatin or euchromatin, is more accessible to the molecules that are going to read the DNA, and so transcription and gene expression can occur here. So a cell can kind of more tightly pack or unpack the DNA in order to help affect what genes are going to be expressed in that particular cell. So let's also talk about another type of DNA that can be found in cells. So in a lot of prokaryotes and a few eukaryotes, you can have these extra bits of DNA. They're circular molecules of DNA called plasmids, and they are outside of the normal chromosome. They were not going to contain usually the genes that are absolutely necessary for the organism to stay alive. Instead, they're going to have some genes that are useful under certain circumstances. So one very common thing that's been researched quite a bit is some bacteria can have genes that let them become resistant to certain antibiotics. And plasmids are often going to be where these genes are stored. And since bacteria can actually transfer plasmids from one bacterial cell to another, they can pass on these antibiotic resistance genes, which makes it really difficult when we're trying to treat bacterial infections. Now, these plasmids can reproduce completely independent of the regular chromosomes as well as the entire organism, and they're on the much smaller side. They'll have a few genes on them, usually anywhere from 1 to 400,000 base pairs. And a cell can have anywhere from one plasmid to thousands of plasmids in it, depending on the particular cell. Now, we've talked about chromosome structure. Let's talk in more detail about DNA structure. So this should look familiar to you. This is a nucleotide. It's the monomer of nucleic acids. It's got 3 parts, a 5-carbon sugar, which in DNA is deoxyribose and in RNA is ribose. It's got a phosphate group. This does not change from nucleotide to nucleotide because phosphates can't be different from each other. And then it's got a nitrogenous base that's also attached. This nitrogenous base in DNA can be either adenine, thymine, guanine, or cytosine, and in RNA it can be adenine, uracil, guanine, or cytosine. Now, inside the cell, in the nucleus and the cytoplasm, these nucleotides usually exist as nucleoside triphosphates, like ATP, adenosine diphosphate, or GTP, guanosine triphosphate, until they're incorporated into DNA or RNA. So you have these free-floating nucleotide triphosphates, and they can get used to produce DNA or RNA molecules. When they're incorporated into a DNA or RNA molecule, they're going to be joined together by what are called phosphodiester bonds between the 5-carbon sugar and the phosphate of the next nucleotide. So the nitrogen bases are going to be really important for us. Let's look in more detail at them. So nitrogen bases are able to be divided into 2 categories. There are purines. Purines have 2 rings that make them up, and they are specifically adenine and guanine. So if you look at this diagram, this is guanine. Notice it has a 6-carbon ring physically joined to a 5-carbon ring. That makes it a purine because it's got 2 rings. Now, one way that you can use to remember this, and I found this on an MCAT preparation website. MCAT is the test you take if you want to go to medical school, is you can remember the phrase pure as gold. The purines are adenine and guanine, A and G. Pyrimidines have only a single ring. It's a 6-carbon ring. Well, it's a 6-sided ring with carbons and nitrogens. Obviously, it's a nitrogenous base. And the ones that are the pyrimidines are cytosine, thymine, and uracil. So one way to remember this is the phrase cut the pie, like pyrimidines, if you want to think that way. And the C and the T are for cytosine and thymine, or you can think there's a U in there for uracil. I just remember that A and G are purines. You can remember pure as gold, and just remember all the other ones are pyrimidines. Now, for base pairing rules, specific bases pair with each other in DNA and RNA. And the rule is one purine is going to pair with one pyrimidine. So you can think of a DNA molecule as being 3 of these rings across. So adenine always pairs with thymine in DNA, or if you're looking at RNA, it's going to pair with uracil. Adenine can't pair with guanine because guanine is another a purine. And we'll talk in a moment about why it cannot pair with cytosine. And guanine pairs with cytosine. Now, these nitrogen bases, pair up with each other by hydrogen bonding. And that hydrogen bonding is what determines what bases can pair with each other. So if you'll look, cytosine and guanine have 3 hydrogen bonds between them. And then adenine and thymine only have 2 hydrogen bonds between them. So that is why adenine cannot base pair with cytosine. They can't form the proper number of hydrogen bonds with each other, because in this place, instead of being able to form a 3rd hydrogen bond, that's a CH bond. That's a nonpolar bond. So that's not going to be able to produce a hydrogen bond. So adenine will not be able to base pair with cytosine. Now, let's talk about RNA real quick. RNA is going to become a very big player, and we're discovering more and more as we research the various processes that happen in a cell that RNA has a lot more roles than we thought it did. So how is RNA different from DNA? Well, RNA is going to have 2 differences in the nucleotides. It's going to have ribose as its 5-carbon sugar instead of deoxyribose. It's very similar in structure. You don't need to be able to memorize the difference between ribose or deoxyribose. They should be labeled on any diagrams you see on the AP test. And then for the 4th nitrogen base, instead of thymine, RNA is going to have uracil instead. RNA is usually single-stranded, but it can actually form some double-stranded sections, so it can hydrogen bond to other pieces of itself as long as there's a matching base sequence. And because it can form these kind of bonds with itself, it can form some pretty complex shapes, and those complex shapes allow it to have different functions. So, how does DNA carry hereditary information? It's all about the sequence of nitrogen bases, the order of the nitrogen bases. You can think of it almost like a language, and just like the order of letters in a language are going to determine words and their meanings, the order of nitrogen bases in a sequence of DNA is going to determine meaning in the DNA. And in this case, we're going to learn later or remember later that it's going to determine amino acid sequences in proteins. The sugar phosphate backbone is just there as structure to help make sure that the nitrogen bases stay in a particular order. So it doesn't have any relevance to carrying genetic information itself. It's kind of like the pages of paper in a book. Those pieces of paper are just there to hold the words, the letters in a specific order to make meaning. The sugar phosphate backbone is just there to hold nitrogen bases in a particular order. So they have meaning. All right, next up, we'll talk about DNA replication, and I will talk to y'all then.